Hello there, welcome to this tutorial on A-level respiration. This will be covered in two parts as there are a lot of content to cover and understanding the concepts takes time. So let's get started. These are the aims of the tutorial: define respiration and briefly outline the types, list the steps in respiration and summarize what happens, list differences between aerobic and anaerobic respiration, explain the differences in anaerobic respiration of yeast and human cell, explain in detail what happens during Krebs's cycle, explain in detail what happens in oxidative phosphorylation, what is chemiosmosis, what other molecules can be used in Krebs's cycle. We'll only go through these in this video. Let's start by understanding what respiration is. Respiration involves production of ATP molecules from an energy source like glucose. There are two types of respiration, which I'm sure you know by now. We have aerobic and anaerobic respiration, where aerobic uses oxygen, while anaerobic doesn't. A point to note is that all anaerobic respiration occurs in the cytoplasm, so no mitochondria is needed. You will be familiar with this equation from GCSE, but now we're going to spice this up a level. Get it? A level, A level. Okay, not that funny, but let's continue. Let's see the steps involved in respiration. There are four key stages. They are glycolysis, which involves the production of two pyruvate molecules from one glucose molecule. Next is the link reaction, where acetyl-CoA is made. This is followed by the Krebs's cycle and oxidative phosphorylation. When we talk about anaerobic respiration in A-level biology, we're only talking about glycolysis and fermentation, which we will see later in this video. Before we get into the process, it's important to understand a few terms that will be used in this tutorial. The first one is phosphorylation, which is the addition of phosphate to a compound. Often this is ADP, so you can make ATP. Phosphorylating compounds makes them reactive. Next, we have oxidation. You should be familiar with this from chemistry, but oxidation can also be described as the removal of hydrogen atoms. Removal of hydrogen is also called dehydrogenation. So dehydrogenation is a type of oxidative reaction. Next is reduction, which is the opposite of oxidation. And hydrogenation, which means addition of hydrogen, is a type of reductive reaction. Next we have coenzyme. To simply put it, is needed for an enzyme to function. Without it, certain enzymes can't. Finally, we have decarboxylation, which refers to the removal of carbon dioxide from a compound. Now there are some important concepts to comprehend. Firstly, all respiration reactions are enzymatic, which means respiration rate depends on temperature. If cells are too cold, very little respiration occurs. Next, enzymes ending in kinases all carry out phosphorylation, that is an addition of phosphate to a compound. And finally, enzymes ending in phosphatases remove a phosphate from a compound. Now, in A-level, books can use different terminology to mean the same thing. This first one is pyruvate, also called pyruvic acid. Next, we have citric acid, which is also called citrate. Next is NAD, which is also the same as NAD+. And finally, reduced NAD, which is the same as NADH. It might seem confusing why two different things mean the same thing, but you just have to get used to it, especially if you want to do medicine. Okay, let's start with the first step in respiration. Glycolysis, also known as glucose breakdown. Glycolysis can be split into phosphorylation and oxidation. When we're talking about oxidation or phosphorylation, it's important to say which compound is undergoing the reaction. For example, here glucose is phosphorylated by ATP to glucose 6-phosphate, which is further phosphorylated into hexose bisphosphate. Hexose bisphosphate is very unstable, so immediately breaks into two triose phosphate molecules, which are oxidized and dephosphorylated to make pyruvate. Triose phosphate is oxidized by NAD+, which itself is reduced to NADH. Two phosphate molecules are removed from each triose phosphate to form 2 ATP. So one glucose molecule undergoing glycolysis produces a net production of 2 ATP, as 4 ATP is made while 2 ATP was used before. and 2-NADH. So glycolysis is described as an anaerobic process, meaning oxygen isn't needed. It happens in the cytoplasm, so it requires no mitochondria. And this is how prokaryotic organisms, which have no membrane-bound organelles, survive. However, there is the question of how or where the NAD comes from to cause glycolysis. And there are two ways. One is lactate fermentation, which occurs in human cells, and ethanol fermentation, which occurs in yeast. Let's look at this in detail. In yeast, the pyruvate is first decarboxylated, that is, carbon dioxide is removed, and then reduced to form ethanol. This allows NAD+ to be made, which is recycled back into glycolysis so that it can continue. In muscle cells, lactate is made. It consists of a single-step reaction where pyruvate is reduced so NAD+ can be made. But a question to answer is what happens to the lactate made? Here's the catch. The reaction is reversible. In order for lactate to become pyruvate, oxygen is needed. This is what we call oxygen debt in GCSE. Remember, lactate is toxic and harmful to muscles as it can cause them to fatigue. The pyruvate made can be used in the link reaction and reduce muscle fatigue. This reaction is catalyzed by the enzyme lactate dehydrogenase, which is also the enzyme that catalyzes the formation of lactate in the first place. Confused yet? This is one of the many reversible reactions catalyzed by one enzyme. You don't need to go into the depth at A level, so we'll just leave it there. Right, the link reaction. The reaction that links glycolysis to the Krebs cycle. It consists of 3 steps. Firstly, pyruvate is decarboxylated, releasing the first CO2 of aerobic respiration, and then it's dehydrogenated, forming acetate. Then coenzyme A is added to make acetyl-CoA. Note this reaction occurs twice, as 2 pyruvate are made per glucose molecule. We'll look into the Krebs cycle and oxidative phosphorylation in the next video. Now this table summarizes the differences between aerobic and anaerobic respiration. Most of them should be straightforward and we've covered all of them in this tutorial. And finally, to finish part 1 of respiration, I've summarized the key differences between yeast and muscle cells. So these are the learning objectives we covered in the video. Use them as questions to strengthen your knowledge. I hope you enjoyed this video and learned something from it. If you did, make sure to like and subscribe, and I'll see you in the next one.